recording for the presentation, and then we will. Okay, so welcome everybody to this month's installment of IMPART's Research Reels, which is a monthly IMPART seminar series. So just to introduce ourselves, IMPART is a national research team headquartered at DMNB, encompassing 45 academic and clinical partners all across 8 universities. And Research Reels is a monthly seminar series that's hosted to improve networking amongst IMPART members, enhance opportunities for future collaborations, and support knowledge of research findings. So we're always looking for more people who want to participate in hosting a research reel, and this is not just limited to PIs. This can be PhD students, postdocs, even master's students, MDs as well, or even med students that are conducting research. If you know anyone or you are someone that's interested in doing a research reel, you can stay tuned for the post-seminar survey, at which time you can nominate yourself or someone else. And that's the same way, you should always look out for our IMPART communications page. So we are all over social media. You can also find us online. So it's another way that you can reach out to us. Send us a direct message or anything like that, or check out our website, our YouTube channel, which is where all the research reels are recorded and uploaded after the fact. And then we're also recently on TikTok. So we're on a new social media platform and we're trying that out. And then, like I said, at the end, the research reels are all uploaded on YouTube. So if you missed one, if you want to go back and watch something else, you can check that out there. And then some of our special events, such as our Celebration of Women in STEM seminar that took place last March, can all be found on YouTube. And without further ado, I'm going to welcome today's speaker. Today's speaker is Dr. Ketul Chaudhary, who is an affiliate member of IMPART and an assistant professor in the Department of Physiology and Biophysics at Dalhousie University. Dr. Chaudhary has a Bachelor of Pharmacy degree from Hemchendracharya University in India. He then moved to Canada to pursue a PhD in pharmaceutical sciences at University of Alberta. And after his PhD, a postdoctoral fellowship at the Ottawa Hospital Research Institute with the University of Ottawa before joining Dalhousie University. And Dr. Chaudhary's research interests and a little bit of what we're going to hear about today include sex differences, right ventricular function, and right heart failure, as well as the pathophysiology of pulmonary vascular diseases and sex differences in stem cell function. His research is funded by Research Nova Scotia New Health Investigator Grant and Dalhousie Medical Research Foundation and Dalhousie University. With that, I'm going to hand it over so we can start watching Dr. Chaudhary's presentation.
Okay, I will share my screen. Can you hear me all right?
Yeah, we can hear you and see your screen.
Okay. Okay, you can see my presentation. And you can see a laser pointer, right?
Yeah, laser pointer and presentation, all good to go.
Perfect. Good afternoon, everyone, and welcome to the seminar. First of all, I would like to thank the organizers for inviting me for the presentation and for the opportunity to talk about research in my my new lab, and also the opportunity to share our new data. So my laboratory studies the cardiopulmonary physiology, and in this presentation specifically, I'm going to talk about sex differences in right-sided heart failure. And towards the end, if we have time, we'll briefly touch the stem cell physiology part. So my laboratory is primarily interested in heart failure. And what is heart failure? Heart failure is a condition as the name suggests, in which heart fails to do its normal function. Now, heart's function is to generate cardiac output and push blood in different parts of the body. So when the heart is unable to do that, the condition is called heart failure. In clinical terms, it is a clinical syndrome where signs and symptoms arise from insufficient cardiac output that does not meet the body's demand. Heart failure is a growing epidemic, and it currently affects around 600,000 Canadians. Every year, around 50,000 Canadians are diagnosed with heart failure, and it causes significant burden on Canadian healthcare system. An estimated cost of heart failure to Canadian healthcare system is around $2.8 billion per year. Now, depending on which ventricle is affected by the condition, the heart failure can be divided into left-sided heart failure or right-sided heart failure. So if the left ventricle is involved, the condition is called left-sided heart failure, and if it is affects right ventricle, then it is called right-sided heart failure. My laboratory primarily is interested in right-sided heart failure. Before we talk about right-sided heart failure, I briefly want to go over the cardiovascular system and talk about circulatory system. So circulation can be divided into two parallel loops or circuits: the systemic circuit and the pulmonary circuit. The pulmonary circuit provides oxygenated and nutrient-rich blood to different parts of the body. And it functions and it offers higher resistance. The pulmonary circuit is connected to the left side of the heart, the left ventricle, and therefore the left ventricle function at higher pressure and resistance. So mean systemic arterial pressure is around 100 millimeter of mercury, and it offers higher resistance. So therefore, the left ventricle function at a higher against higher pressure and resistance. So left ventricular systolic pressure is over 100 millimeter of mercury. Now, if we talk about the second loop, which is the pulmonary circulation, it offers much lower pressure. It offers much lower resistance, and therefore the right ventricle, which is connected to the pulmonary circulation, it functions at a much lower pressure and against much lower resistance. So if you can see here, the mean pulmonary artery pressure is only 50 millimeter of mercury compared to 100 millimeter of mercury in the systemic circulation. And if you look at the left ventricular systolic pressure, it is over 100 millimeter of mercury, while right ventricle, it ranges up to 25 millimeter of mercury. So normally, right ventricle function against much lower pressure and resistance. However, under certain conditions, the right ventricle can be exposed to higher pressure and resistance, which can have adverse effect on the right ventricle. So under normal condition, right ventricle function as at a much lower pressure. However, in this under congenital heart disease or in response to pulmonary hypertension, it can be exposed to higher pressure. So in congenital heart disease such as Eisenmenger disease, which involves ventricular septal defect. So there is a defect. So there is a wall between left and right ventricle, and if there is by birth there is a defect in this this wall, then blood can go from one ventricle to another ventricle. Normally, the pressure in left ventricle is higher, so blood will enter from left ventricle to the right ventricle, and that will create increase in pressure in the right ventricle. Now, the other condition is pulmonary hypertension, which can be caused by multiple reasons. One of them is pulmonary vascular disease, so the disease of lung blood vessel. So if there is a disease of lung blood vessel, which results in loss of blood vessel in the lung, and it increases blood pressure in the lung. Now, if there is pressure increase in the lung, it will also increase pressure in the right ventricle. Similarly, if there is left-sided heart failure, the left ventricle is not able to push blood into systemic circulation. Now that flows into the lung and causes pulmonary hypertension. And this increase in lung blood pressure then again increases pressure in the right ventricle. So due to congenital heart disease or pulmonary hypertension, either due to pulmonary lung disease, pulmonary blood vessel disease, or due to left heart disease, it can increase pressure in the right ventricle. Now, initially, when right ventricle experiences this increase in pressure, it undergoes remodeling, and initially it is an adaptive remodeling, where to handle this increase in pressure and push the blood against this higher pressure, the right ventricular wall gets thicker. So there is increase in thickness of the right ventricular wall. However, the cardiac output, which is the amount of blood that is pushed by the lung, pushed by the ventricle in per minute, it is maintained, and the chamber size of the ventricle is maintained. However, if this condition persists, or this higher pressure persists for a longer period of time, or if there is if the patient is genetically predisposed or susceptible, then the RV undergoes a different type of remodeling called maladaptive remodeling. Now, in this situation, the cardiac output or the function of the right ventricle declines, and you can see the right ventricle gets enlarged. And this maladaptive remodeling ultimately leads to right-sided heart failure, and the symptoms arise, which includes fatigue, peripheral venous pressure increase, accumulation of fluid in different parts of the body, and ultimately this condition leads to multi-organ failure and death of the patient. Now, why are we focusing on the right ventricle, and why right ventricle is important? So some studies have shown that right ventricular function is important for survival of patient. For example, patient affected by heart failure with preserved ejection fraction. This is a type of left-sided heart failure, and this leads to increase in this leads to pulmonary hypertension and increase in right ventricular pressure. So in these patients, if you look at this graph, the green line shows right ventricular ejection fraction. Now, right ventricular ejection fraction is a functional measure for right ventricle. And if the right ventricular ejection fraction is more than 49%, these patients are presented by green line. And if the ejection fraction is less than 49%, then they are presented by the red line. And you can clearly see that if a patient with better right ventricular ejection fraction and therefore better right ventricular function, they have better survival compared to the patient who have poor RV function. Another study which studied the pulmonary arterial hypertension patient. This is the pulmonary vascular disease that causes pulmonary hypertension. So in these patients, again, right ventricular ejection fraction of more than 35%, which are presented in blue line. So in these patients who have better right ventricular ejection fraction and right ventricular function, they have better survival compared to the patient with poor right ventricular ejection fraction. This study also looked at this data from another point of view, and if you look at this graph, which compared the patient who had stable or increasing RV function compared to the patient who had decreasing RV function. So even if the patient had lower than 35% right ventricular ejection fraction, but if they had stable and maintained right ventricular ejection fraction and maintained right ventricular function, then they had excellent survival compared to very poor survival of patient who had decreasing right ventricular ejection fraction and therefore deteriorating RV function. So if you interpret this graph differently, then you can say that if you stabilize RV function, so if you prevent deterioration of RV function in these patients, so the red line, if you prevent deterioration of RV function in these patients, you can actually improve survival. So therefore, maintaining RV function is very important for patient survival, and preserving RV function can improve patient survival. However, there are some limitations. So we have some therapies available for left-sided heart failure, and these conventional therapies have proven benefit in left-sided heart failure. However, these are not effective in right-sided heart failure because of obvious reasons. The shape, size, structure, and even function of both the ventricles are different. As I mentioned earlier, both function against, both are connected to different types of circulation, and they function against different resistance and pressure. Even their embryological origins are very different. So there are key differences between left and right ventricle, and therefore therapies that are effective in left-sided heart failure are not effective in right-sided heart failure. There are no specific right-sided heart failure therapy for these patients. And therefore, to help these patients, new therapies are needed, and better understanding of pathophysiology of right-sided heart failure is needed to find these novel therapies. Now, studies have reported important sex differences. So the female RV functions differently than male RV. A study by Ketul et al. In 2011 showed that irrespective of ethnic background, females have better right ventricular ejection fraction and therefore better right ventricular function compared to their male counterparts. And this is under normal, healthy condition. And this is also true in response to a disease condition. So if you look at this graph, in the middle graph, this graph represents pulmonary arterial hypertension patient. And in these patients, females have better right ventricular ejection fraction and right ventricular stroke volume, both markers for right ventricular function. And females have better right ventricular function compared to their male counterparts. And this translates into better survival in female patient compared to male. So we know from previous slide that patient with better right ventricular function have better survival, and female have better right ventricular function, and they have better survival. However, we don't know the mechanism of these differences. And if we understand these mechanisms, we may be able to help the susceptible male population. At the same time, the similar mechanisms can be used to treat female patients who are susceptible. To study the mechanism, we use animal models in my laboratory. And a previous paper, and we use FISCHER CDF rat in our studies. In my previous paper that I was involved in, has shown that FISCHER CDF rats are right heart failure susceptible rat strain. So in this paper, we used an animal model of pulmonary hypertension, which is a gold standard model to study pulmonary arterial hypertension, which is a type of pulmonary hypertension. And in this model involves injection of a drug called Sugen or SU, which is vascular endothelial growth factor receptor 2 inhibitor, and that causes pulmonary endothelial cell apoptosis, pulmonary vascular endothelial cell apoptosis. So the cells that make the lining, innermost lining of the blood vessel, they're susceptible to this drug, and this drug causes apoptosis or death of those cells. When it is combined with chronic hypoxia, so low oxygen environment, then it induces progressive pulmonary hypertension in rats. So combination of Sugen and hypoxia induces pulmonary hypertension and increases right ventricular pressure in both the rats. And you can see when the S represents SD rat and F is for FISCHER CDF rat. And in both these rats, Sugen hypoxia induces similar increase in pressure, in right ventricular pressure. Despite similar increase in right ventricular pressure, and in this study we also showed their lung disease was similar. So similar lung disease, similar increase in right ventricular pressure. And despite that, SD rat showed excellent survival. So up to 8 weeks, there was no death in this model. However, in Fisher CDF rat, if you look, none of these rats survived post day 40. When we studied the mechanism, we found that Fisher rat had very poor cardiac output compared to SD rats, and they had increase in chamber dilation, and they had other signs of maladaptive RV remodeling, suggesting that despite similar increase in right ventricular pressure, SD rats develop adaptive remodeling and maintained RV function, and they had better survival. Whereas Fisher CDF rat developed maladaptive RV remodeling. They had reduction in cardiac output, increase in chamber dilation, and they showed poor survival in response to increase in RV pressure. So, and we concluded that Fisher CDF rats are susceptible to right-sided heart failure. So we decided to study sex differences, we decided to use Fisher CDF rat in our studies. Now, we suspected that sugen, which causes the lung endothelial cell apoptosis, it might be influenced by female sex hormone, and therefore we decided to use a surgical model, which is called the pulmonary artery banding model. So in this model, we open the chest of the animal, we put a suture under the pulmonary artery, and we place a needle beside the pulmonary artery, and we tie the suture completely around the vessel and the 18-gauge needle, and we immediately retract the needle. What that does, it leaves the opening similar to the size of 18-gauge needle around the pulmonary artery. So it causes a fixed constriction around the pulmonary artery. And because of that, there will be a pressure increase in the right ventricle. So we do this surgery in Fisher CDF male and female rat. And then at 1 week and 2 week time point, we perform different measurement, which involves catheterization. So we put a catheter into the right ventricle to measure the right ventricular pressure. We perform echocardiography to study cardiac structure and function. And we collect tissue and perform histological analysis to study structural remodeling. So in response to pulmonary artery bending in male and female rat, both group developed increase in right ventricular pressure at 1 week and 2 week time point. However, there was no difference between male and female rat. In response to increase in right ventricular pressure, we see increase in right ventricular hypertrophy in male and female rat at 1 and 2 week time points. Now, despite similar increase in pressure, we observed that cardiac index, which is the cardiac output divided by the weight, which is a functional marker for the right ventricle in these patients, in these animals, and we see that females have better cardiac index compared to their male counterparts at 1 week and 2 week time point. Fractional area change, which is a surrogate measure for ejection fraction, which is measured by echocardiography, and we see that fractional area change decline in male rats subjected to PAB, and we observe better fractional area change in female rats subjected to PAB at 2 week time point. Now, if you look at the structure, we observe progressive increase in right ventricular internal diameter in male rat. However, this change was not present in female rat subjected to pulmonary artery banding. We also saw a progressive increase in cardiomyocyte cross-sectional area, and this progression was slower in female rats subjected to pulmonary artery banding. So together, this data shows that in response to pulmonary artery banding, female rats develop adaptive RV remodeling where they have maintained cardiac output, RV function, and chamber size, whereas male rats develop maladaptive remodeling, and they show reduction in cardiac output and other functional markers, and show increase in RV dilation. So we kind of have, in this model, we mimicked the condition that we saw in in patient data. Next, we wanted to study the mechanisms. And in our previous study, we observed differences in vascular density between ST-RAT and FISCHER-RAT. Again, I want to remind that ST-RATs are the model of adaptive RV remodeling, whereas FISCHER-RATs are markers for mild adaptive RV remodeling. And FISCHER-RATs are susceptible to right-sided heart failure. And in this case, we looked at blood vessel density in the right ventricle using CD31 staining, which is a marker for endothelial cell or the endothelial cells. And we also performed fluorescent microangiography, which involves injecting or casting the vessel in the right ventricle using agarose gel, which is mixed with fluorescent bead. So here we are measuring the density of the vessel. Here we are measuring the volume of the blood vessel in the right ventricle. And in both cases, we observed that female with the Fisher CDF rat had lower endothelial cell density and volume compared to ST rat when they were subjected to pulmonary hypertension. And this data showed that right heart failure in Fisher CDF rat was associated with reduced vascular density and volume in the right ventricle. Similarly, another study has shown that in pulmonary hypertension patient who have maintained cardiac function, they have preserved CD31 and VWF staining. Now, both of these are marker for endothelial cell. So this shows that patients who have maintained right ventricular function, they can maintain or they have preserved vessel density in the right ventricle. However, patients who do not have preserved vascular density, they have deteriorating RV function, and they develop maladaptive RV remodeling and develop right-sided heart failure. So both these data show that maintaining right ventricular vessel density is important to prevent development of right-sided heart failure. So to see if similar mechanism was present in our animal model and if similar mechanism was involved in the sex differences, we quantified endothelial cell in the right ventricle of female and male rats subjected to pulmonary artery bending at 2-week time point. We used the same marker that was used in the human study, and we saw a higher density of endothelial cell in the right ventricle of female Fisher CDF rat that were subjected to PAB compared to the male rats. To study the mechanism, we perform gene expression analysis. So under naive gene expression analysis of the endothelial cell, and here we use PCR array. And we looked at 84 different angiogenic genes in the RV of male and female rat. And under naive condition, we did not see a major difference between male and female RV. However, in response to pulmonary artery bending, we observed a rightward shift in these genes, which shows that these genes, majority of angiogenic genes, were highly expressed in the right ventricle of female rat. From there, we identified 4 candidate genes, and one of this gene was sphingosine kinase 1. Now, this is an enzyme that can catalyze the reaction of conversion of sphingosine to sphingosine 1-phosphate. Sphingosine 1-phosphate is, or S1P, is a bioactive lipid which circulates in blood, and it binds to various receptor, and binding of it to receptor produces angiogenesis, which promotes vessel growth, and binding of this bioactive lipid to another type of receptor produces anti-angiogenic effect, which inhibits the blood vessel growth. In the heart, endothelial cells, or in endothelial cell, this particular receptor is the dominant one, so it is highly expressed. So binding of this receptor to the binding of the S1P to this receptor would produce increase in vessel growth. Interestingly, a previous study has shown that SPHK1, which produces S1P, it mediates the estradiol-mediated angiogenesis. So in this study, the authors performed in vitro angiogenesis assay. So this is also called the matrigel network formation assay. And in this assay, endothelial cells are plated on this matrigel matrix where they form networks. And this complexity of this network is suggestive of their in vivo angiogenic ability. Another assay they performed was migration assay. So this is a scratch wound migration assay where a monolayer of this cell is formed and a scratch is produced. And with or without treatment, you monitor the movement of these cells and closure of this gap by these cells. And more closure is indicative of higher migration of this cell, and more higher migration is indicative of the better angiogenic potential in vivo. So in both these assays, the author showed that in response to estradiol treatment, these cells form more complex network, and this network formation was inhibited by deletion of the receptor for S1P. Similarly, migration was increased in response to estradiol, and this effect was inhibited or reduced by inhibiting the S1P1 receptor. And the author concluded that estradiol through SPHK1 and S1P1 increases blood vessel formation. So to study if similar mechanism is present in the right ventricle, and it is contributing to the sex differences in the right ventricular vessel density and therefore right ventricular function, we isolated right ventricular endothelial cell from Fischer CDF rats, and this data confirms the purity of these cells. The majority of the cells that we isolated express the CD31, and these are a pure culture of endothelial cell. And when we treated plated these cells on matrigel and treated them with vehicle or estradiol, we saw more complex network formation with estradiol treatment. We quantified this. So in response to estradiol treatment, we see more complex complex network formation. And this network formation was inhibited when, with the inhibitor of SPHK1, suggesting that estradiol can increase angiogenic ability of the right ventricular endothelial cells, and it is mediated by the sphingosine kinase 1. This is where we are with this study. So far we have shown that female Fischer rats demonstrate adaptive RV remodeling, which mimics the human condition. And the better RV adaptation in female Fischer CDF rat was associated with increase in angiogenic genes, which will promote blood vessel formation in the RV. And one of these genes is SPHK1, and we have shown that it is involved in a female sex hormone mediated increase in blood vessel forming ability of right ventricular endothelial cells. We are validating these mechanisms in vivo right now. And briefly, I want to go over our stem cell research. So we are interested, or we are working on characterization of cardiac stem cell antigen 1 expressing cell. And this is our NSEC research program. So stem cell antigen 1 or SCA1 has been used as a marker for stem cell in various organs. And in the heart, initially it was suggested that it is a marker for cardiomyocyte stem cell, or the cells that give rise to cardiac muscle cells. However, recent studies have shown that indeed these cells are not the precursor for cardiac muscle cell, but indeed they are the precursor of endothelial cells. So you can see here the SCA1 cells are marked by green, and PCAM, which is CD31, which is an endothelial cell marker. And you can see all the green cells are also expressing the endothelial marker, suggesting that all the SCA1 positive cells are also endothelial in nature. The study also showed that these cells do not express any cardiomyocyte marker, so they do not form cardiac muscle cells. This data again confirms that they may be endothelial cells but not cardiac muscle cells. So, however, this study did not perform a global analysis. So they looked at, they performed the staining in sections, and they did not perform a global analysis, as well as they have not done a functional analysis of these cells. And that's where we started working on these cells. And so in my lab, we are using flow cytometry to study perform global characterization of these cells, and also isolation and functional characterization of these cells. So for that, we collect the heart, we separate left and right ventricle, we digest the tissue, then we obtain single cells, and we perform staining with specific marker for endothelial cells and stem cells, and analyze by flow cytometry and characterize these cells. So this graph here shows that in 4 quadrants. So in the bottom right quadrant, the cells in this quadrant show CD31. So these are endothelial cells only. On the top left corner, the cells would only express SCA1, which is the stem cell marker. So these are the stem cell marker, but they are not the endothelial cell type. And if the cells are present in this quadrant, then they express the SCA1 and CD31, meaning that these are stem cell, and they have endothelial markers. And this is double negative population. So you can see that any SCA1 cell or any cell that is expressing SCA1, they're also expressing CD31. So majority of these cells are endothelial in nature, which is consistent with previous finding. We went on and performed functional characterization. So we did acetyl-LDL and lectin binding. Both are the characteristic of endothelial cells. And we compared acetyl-LDL uptake and lectin binding in the SCA1 expressing cells and the mature endothelial cells. So in these cells. And the SCA1 expressing cell also take up acetyl-LDL and bind to lectin, similar to the mature endothelial cell, suggesting that these cells are functionally endothelial-like cells. We next isolated these cells and performed the network formation assay, and endothelial cells, when plated on matrigel matrix, they form these network, and this data again confirms that these cells are functionally, the SCA1 positive cells from the heart are endothelial-like cells. We also looked at abundance between left and right ventricle, and we see a higher abundance of these cells in the right ventricle compared to left ventricle and septum. This is particularly interesting for our research, and we are currently working on further characterization of these cells and looking at their involvement in right ventricular remodeling. So I'll stop there, and first I would like to thank my trainees. Without their hard work, this would not have been possible. So all credit to my trainees, my past members, Erica, Krishani, who are master student in my lab, recently defended their thesis. My research technician, Sheetal Chaudhary, and Tony, and my honor student. I would like to thank my collaborators, my postdoctoral mentor, Dr. Duncan Stewart, who supported immensely during the start of this work. Other collaborators, Dr. Ankit Desai, Dr. Alex Queen, who is also a mentor and a collaborator for another project. Dr. Thomas Pullin, we are working together on the right heart failure and sex difference studies. And I'm collaborating with Dr. David Horn and Christian Herman for the stem cell characterization part. And finally, thank you to all the funding agencies, particularly Research Nova Scotia, DMRF, and Delcru. Without their support, I would not have been able to do the work. And support from IMPART, CFI, and Dalhousie University. So thank you, and I'll be happy to take any question you may have.